my pleasure to welcome Albane to visit us today and give it actually the first seminar for this quarter. I mean, the first one was postponed due to bad weather. This one was borderline, but she was already here. Sorry. So Albane did her undergraduate in France, and then she did her PhD with Eric Lauger in the Department of Applied Math and Theoretical Physics in Cambridge, which is basically the mecca for fluid mechanics, the place to do fluid mechanics. And after that, she started the postdoc. She's doing now postdoc she's a Simons-supported postdoc in the University of Pennsylvania, where she's doing research in the broad field of mathematical biology. And she's going to tell us something about microswimmers. So, welcome.
Hi, everyone. Thank you very much for having me here. Thank you, Petia, for the invitation and introduction. So today I will be talking about, microswimmers and complex environments. So complex environments meaning realistic settings, both for experiments and for biological questions. I am in a math department, but this talk will be very applied. I tend to work in collaboration with experimentalists, and everything today will be results of these collaborations. This thing worked. We're gonna, oh well.
That's why the old-fashioned way.
Oh no. Oh, okay. That works. Okay, so when I'm talking about microswimmers, a lot of the time I'll be talking about biological microswimmers. So that can range from bacteria, which are about a micron in size, to bigger eukaryotic cells like spermatozoa, and also zooplankton, algae, and things like that. Now you can also have artificial swimmers. These artificial swimmers, there are two broad classes. One that are really biomimetic. So your bacteria propel with helical flagella. Well, you just stick a helix to some head and you rotate it with a magnetic field, and your swimmer is moving. Or you can have a different class, which are, for example, relying on chemical gradients or things like that to self-propel. So a big thing with microswimmers is that we understand fairly well how they're moving when they're in the bulk and when there's nothing else. But of course, if you want to use these artificial microswimmers for any biomedical applications, and if you want to understand the swimmers in their real environments, where of course they're going to interact with other swimmers, with boundaries, and with biological fluids, you have to look at more complex and realistic environments. So this is a very simplified picture, but one thing that you're going to find in these environments are boundaries. So you're going to have just plain, simple boundaries, but you can also have some very weird confinement. We'll come back to that. And this is very important. If you just take E. coli, a bacterium, when it swims in the bulk, it's swimming in a straight line. You put it near a surface, and it's going to be near surfaces a lot because it bounces into them, but also because it's attracted to them hydrodynamically. Well, it starts swimming in circle. And this is because the propulsion mechanism, the helix, is chiral, and you have a rotation of the helix, counter rotation of the head, and both of these induce a drift, and this makes clockwise circular trajectories. We'll also come back to these circular trajectories. The swimmers are also not just going at random. They're mostly swimming for mating, for nutrients, to escape predators. So of course, they're going to respond to signals in their environment. They can do that actively, so get a signal and have a biological response to it, but they also very often do that passively because they don't have neurons for most of the smaller organisms and things like that. So you can have external fields. They respond to magnetic fields, electrical fields, flow fields, and they also respond to light. And finally, the last thing is that they're swimming usually in, or very often, in biological fluids. So you have suspended objects. These can be rigid particles, which are already a bit of a complication, but they can also be deformable particle polymers. And then you have elasticity, changes in viscosity. And you also have interactions with the obstacles themselves when the obstacles are big enough compared to the swimmer. So, for example, if you're thinking about microswimmers in the body, you're going to have saliva, which is a complex fluid. You're going to have mucus. You have blood as well, which has big cells inside. And the big idea of my work is to try to understand in very specific setups how these one or more of these effects are going to affect a particular situation. So very often this means also, of course, the effects become very different when you have many swimmers instead of one. So you start having collective effects and things like that. In this talk, I'll only be talking about having one swimmer. So a big issue is to find a compromise between having something that's very specific to a given problem, a given biological question, a given experimental setup, and having techniques that are fairly general, or thinking about ideas that you're going to be able to reuse a bit more systematically in different setups. And so today, I'll be talking about 3 different problems. Hopefully all the slides, except the titles, are in the visible part. I'll be talking about 3 problems. So ideally, I want to introduce sort of degrees of complexity. I will introduce boundaries first, and I will introduce complex fluids. And then the third problem is about what happens when you have boundaries, complex fluids, and also an external flow. In all cases, again, these are results from collaboration. And in our first problem, we're just going to consider boundaries, but very specific ones. So you've maybe seen things like that. Sometimes on the seaside, you have normal foam formation, and sometimes you have these massive foam eruptions where you have it all over the sand or all over a lake. These are very often the result of some pollution. It can be directly because there is a pollutant that acts as a surfactant and creates the foam, or it can be because there is a bloom of a microorganism that feeds on the chemical that is causing the pollution, then explodes in terms of population and forms surfactant, which is the case here. And the question that my collaborators have been addressing is what happens to the ecosystems when there is massive foam formation like that on a body of water. And so this is not the answer to everything, but Florence Elias in her group, they've been working on what happens if you put motile microorganisms inside foam, and you look at the foam draining because of gravity. So initially it's very full of fluid, and then it's just drying up, and the fluid is just on the bottom part draining by gravity. And the question is, does it make a difference if the algae inside, which look like this, Chlamydomonas, are motile or not? And the intuition you might have is that if you're motile, you're going to be better at escaping the foam. But they found exactly the opposite. When you put the mix of microorganisms and liquids, you make it a foam, you wait for it to drain, the live algae actually get trapped. At least half of them just get trapped here and it just, well, die there. And the question that they ask is, why? So if you want to think about foam, most of it is air. And then in between 2 bubbles, you have a very thin liquid film with surfactant. And that's too thin for having any swimming in there. And all the plankton, all the microorganisms are going to be in the regions between 3 bubbles. So you have 3 circles that are intersecting, and you have these shapes, which are called plateau border, which are basically tubes that have these 3 circle shape. And all the flows and all the swimming are going to happen in these plateau borders. And this is actually a complex problem because you have a mix between the flow, you have how the microswimmers respond to gravity, and you have their interaction with these weird structures. And to sort of take a first look, they created a 2D microfluidic chamber that looks like the cross-section of one of these plateau borders. So it's twice as high as Chlamydomonas, which it doesn't have tens of cilia, it just beats them in a breaststroke fashion. And we're just tracking the chlamyd inside that plateau border. So this is the tracking that I did from their videos. The big idea, if you look at that, initially I was really looking for hydrodynamics because this is what I do. But if you look at that, you kind of see that it's just really something that's going in a straight or noisy trajectory, bouncing on the wall, and then just moving away. And then very often it does one of these things. It just stays trapped in a corner. And so we started modeling this. And So the first thing is, if you look at the spatial probability distribution function, they do spend about a third of their time in the corner, and they also spend more time at the boundary. And so this is the features that we were trying to reproduce. And well, initially I started looking into hydrodynamics, but then it turns out that the right way to see it is really the steric part of the contact. So if you think about a microswimmer and a wall, there are two ways to see the contact. The first one is purely hydrodynamic. There is no contact, and the fluid, because of the no-slip boundary condition, is going to be affected by the presence of the swimmer. The other way is there is a contact, and really what happens is between the swimmer and here the flagella and the wall. So this is a movie from Ray Goldstein's group. And there were further experiments that really showed that they were, I think I can try doing that a bit more. Okay, so there really are two types of contacts when you think about clammy, and the most frequent one is the steric one. So in the steric one, the algae arrives at an angle, hits the wall, and then leaves the wall at an angle that does not depend on its previous trajectory. It's basically a constant departure angle. Then there are hydrodynamic interactions where the clammy does not hit the wall, it gets reoriented because of the flow, and leaves with an angle that's the same as its impact angle. But here we're mostly seeing the first type of contact. So what we can think about for the system, the most basic toy model you can do is really a 3 circles billiard. And then you take all the complexity of the foam and you just move it to a lot of trigonometry. So if you have a trajectory, it's going to look like that. But you can really just do, so this was during lockdown number 2 or something. So good time to do trigonometry. You can start from any point at the lower circle, and then you can look at where it's going to go at the next point, and then 1 point further. And from that, you can get orbits, 3-point ones. You can get 6-point orbits, yes.
How does way you leave it a constant angle from the steric contact? I would guess that would be reflected too.
No, so it's not really obvious, but if you look at the video, it's really because the flagella, what happens is it's going to spend a bit of time at the boundary, and one of the flagellum is going to be stuck, mostly, and it's the time that it's going to take to reorient, and as soon as it hits the right angle to go away, then it goes. So it's really like that angle. And so
this is the size thing, right? So it's the size of the curvature.
Yes, but we think that, so here we had that question for, so there are models with smaller obstacles, which are typically the same size as the swimmer. And in that case, the angle is going to vary depending on the curvature. Here, it's big enough that we don't really see any difference. We tried different curvatures, and the distribution of out angle is mostly the same. But it is noisy, so I'll come back to that. So this is without noise, you just have that constant scattering and you look at the trajectories. And basically you can get stable orbits, but most of the time you're going to have trajectories that end up in the corner. That's not really a surprise. If you have a cusp like that, it's an attractor. So the size of the attractor depends on the bouncing angle, but roughly you're going to get there. And so you can do a phase map of the whole system. So you really have 2 parameters. One is the first, where you start your trajectory, and the other one is at which angle you're going to leave. And if you look at these, the colors just mean that you're getting attracted to one of the corners, so red, blue, yellow. And the black ones are trajectories that are actually not trapped. So it's the clammy that are going to survive. And you see that there is not many of them. It happens if they have a very big bouncing angle, and then the attracting region is basically 0. And it happens for these very rare periodic trajectories. Except that if you put noise in the system, these periodic trajectories don't exist anymore. And generically, you're going to get to the corner, and then it all depends on how fast you can escape a corner, which is basically just an exponential distribution on how fast you're rotating upon yourself. And so you can take a distribution for the bouncing angle that basically mimics the experimental data, and then you can take that exponential time to exit the corner, and basically that's all there is to that system. You can reproduce quantitatively. So these are the theory and these are the experiments, and you can see that you can mimic very well that trapping effect.
Is this chaotic?
I don't think so. People have done this type of constant scattering billiard inside polygons, and then you can get some chaos. Here, I think because of the attractor region, unless you're at pi/2, you don't have chaos, but I'm not sure.
Billiard table and
Yeah.
it's not exactly at the right angle, that's a chaotic system.
Yes, absolutely. But I don't think it happens here because of the cusps that are attractors and because of the constant scattering angle.
But the black region that you were showing, that's also not chaotic.
No. Yeah, so it's kind of a nice system and you get the basic feature. And that's a good explanation for why the microorganisms are actually trapped there. We're not sure it's the right one, and they've been moving to doing 3D experiments where they have some flow and they can look at whether this really applies in 3D columns. We're waiting for the results and we'll work on that. But yeah, yes.
So you mentioned these experiments with the dead algae versus live algae.
Yeah.
So why would being in the corner cause you to get?
Oh, yeah, good question. I should have said that. If you think about the fluids flow, if you're in this corner, because you have no slip boundary conditions at the wall, there is basically no flow in the corner. So if you're there, you could be physically stuck because you have very thin boundaries, but you could also just not be experiencing any downward flow. And if you're swimming, you might also be swimming up. So that's a combination of that. I don't exactly know which is which is why we're looking at 3D. But basically, the flow is weak enough so that they could be swimming up in these regions. And then, of course, there's the question of what happens if the plateau is horizontal. And yeah. But roughly, it's that there's no flow in corners. Okay. So this is really a system where you have hydrodynamics that are really important, but they're completely, the only thing that they determine is how fast the algae is swimming, and the structure, the probability of being anywhere is really driven by steric interactions. Now, the second thing that I want to talk about and that I mentioned is complex fluids. So complex fluids are a very big field. It ranges from a lot of people in engineering are really trying to quantify what is the rheology of a given fluid, and that's a big question. So when you have any mix of things, just characterizing the response of your fluid to any type of stimuli is very difficult. And if you add to that the biology, there were a lot of experiments, for example, of E. coli, and it appeared to swim much faster in polymeric fluid, but it turns out it was eating the polymers, and so it was going faster because it had more food. But also it was going faster because of the polymers. So, but there are biological questions, there are mechanical engineering questions, and then there is the mathematical question of how do you model this and the interplay between the two. And so the idea of this project, which was done, the experiments were done in Roberto Zenit's group at Brown by his student Andres Zambrano. And the idea was to have a minimal controllable thing. So for the fluids, you take very viscous glycerin and you add hard spheres. So that's basically as easy, like that's as simple as you can get for a complex fluid. And then for the swimmer, you get an artificial swimmer where you have a rigid helix and you're just controlling how fast it's rotating. And what you're measuring, basically, you're inputting omega, so the rotation rate, and you're measuring the speed at which your object is moving.
How big is that?
Centimeter size, but it's still very low Reynolds number, very viscous system. So what they got here on the swimmers experiments directly is that if you add more spheres, but staying at the dilute volume fractions, the swimmer goes faster. So it's not necessarily an obvious effect, and I'll come back to that, but if you take these suspensions and you just drop an object that's going to sediment, it's going to sediment slower, always, if you have a suspension, and that just comes from the fact that Stokes flow, so the flow without the spheres, is the one that has minimal dissipation. You cannot do something that has less drag than a Stokes flow. And therefore, if you add spheres, it's always going to have more dissipation, so more friction, so it's going to be slower. But your helix is going faster. It doesn't, so it's going in that direction. That's because the data is hard to get. And that's why I'm going to move to the second experimental system. That's exactly that. Because actually your swimmer it's really hard to do density matching for both the spheres and the swimmer. And if your swimmer was wiggling a little bit, then it's hard to track. And also it's hard to have the right fields, and it's hard to image because you have a rotating magnetic field. So all of that means that there will be some wiggle, and there will be some wiggle on that experimental setup too. But the idea here is instead you're just looking at the helix, you're rotating it, again, constant speed. But this time the output is going to be the force, which is basically the thrust, and the torque, which is basically the drag. And so this one is a bit easier to do the measurement with. So they're still noisy also because of the printing of the helix, because of the systems and so on. But this is a bit of a more controllable system. And so, as usual, I only do the theory. And so my question is, well, what happens to a helix when it's rotating, and how fast is it going to move? And if you want to go back to the first question, it's really, why does a helix move in a fluid if you rotate it? And one way to see that is to split your helix into tiny cylinders. And you're going to do a force balance on all of these cylinders individually, making the huge assumption that these cylinders are not interacting with one another. But then your question becomes, I have one cylinder, I impose a force that's not in the parallel or perpendicular direction, and I want to know where my cylinder is going. And that's much easier because you can just split it into the parallel and perpendicular direction. You can compute the drag coefficient for both this motion. And fairly intuitively, it's harder to move a cylinder in that direction than it is in this direction. So if you go back to the sketch in the middle, it means that you're going to have a drift. If your cylinder is dropped like this, it's going to drift like that. And now you put all these drifts from the cylinder into the geometry of the helix. They all go in the same direction. So that's the trick. Really, a helix is about exploiting this difference in drag coefficients, which is basically it's twice easier to move like this than like that. And you're exploiting it in the geometry, and your optimal geometry is going to come out from that really drag coefficient difference here. So this is called resistive force theory, and it's the easiest way to understand why your helix is moving. And the question was, how does that setup fare when you have a suspension? There was one approach in the literature before, which was basically to consider the suspension as a fixed force medium, so as a fixed matrix. It turns out that it doesn't work for our experiment. It's like way off. Even for a theoretician, that's like not good. The reason for that is that if you're considering a fixed matrix, it's not responding to your flow. So it's really a different type qualitatively of interaction between a wall and something that's floating around you. So really the floating part, the fact that you're force-free and just moving with the fluid, is key here to understanding that interaction. So what I did is I took exactly the opposite approach. Instead of having a matrix, you have one sphere and you have one cylinder, and you just look at that system. And my question now is, I have my cylinder that's sedimenting like this. I put a sphere here. Is it going to drift more or less? If it drifts more, then my helix is going faster. And if it drifts less, my helix is going slower. And so all I have to do is just look at the hydrodynamic interaction between my cylinder in this direction and the sphere. And that I can do easily. I need to know what the flow from the cylinder is. And a classical result is that you can, to the first order, just approximate your cylinder as the line of point forces. So basically, it's going in one direction, and so it's imposing a force everywhere where it's moving. You have a line of flow length, you can integrate that, and you get the flow that the sphere is feeling. Now, the sphere is going to be moved by the flow, but it's also going to create a disturbance flow. And that disturbance flow is well approximated by a stresslet, which is, again, a solution of Stokes' equation. But this time, the idea is to have a force-free solution. So it's the Stokes sets are the grain function. It's really the first order thing. If you put two Stokes sets side by side in some configuration, you get a stress set. So this one is the force-free solution that you build from your grains function. And so you can do integrals. They look like this. And I sort of discovered Mathematica, but only halfway through the calculation. So some of this is done by hand. Yeah, I know. Sad. So you get these results. They're not super friendly. The main thing that you can see is that you have different symmetries in the problem for this one and for this one. One is axisymmetric, the other is not. So that's what you get there. Yeah. Yeah, so I did the calculations for this. I'm going to ignore that. But so you have similar type of results, except they go in opposite directions. But for the helix, I'm going to ignore that, but that's a really good question, actually. So to understand what happens to a helix, I'm going to assume that there are spheres, but only in that part, because you're going to put the cylinders one by one, and you don't really have end effects, except at the tips. But if you put the sphere at the wrong place, then it will go in the opposite direction, actually. Your cylinder will drift less. But here, I'm going to say that the spheres are only there, because I'm, again, thinking about a helix. And so you can also do the integral, and you can find the first order perturbation to your drag coefficients. And again, some expressions. So some changes in coefficients which are just geometry. And then the big thing is, it's really simple. If you're moving that way, you're creating a bigger flow. And therefore, your disturbance flow is also going to be bigger. And therefore, the effect of the sphere is bigger. And therefore, you're just increasing. It's a self-sustained thing. So it's really that simple. And then geometry. But it works. If you use that drag coefficient and you look at what happens in the experiments for the optimal helix, you get a qualitative match. And I'm not claiming quantitative here because resistive force theory is very hand-wavy. And actually, if you change the geometry of the helix, yeah, you can get anything. But the trend is there and the order of magnitude is there as well, which is already an improvement compared to the porous media. And now if you want to move to really understanding what happens with the geometry, you cannot just deal with cylinders. Of course, you have to deal with the helix. And you can do exactly what I did with a cylinder, but with a helix. So instead of having your singularities that are placed on the cylinder, you can do singularities on a helix. So this is basically the solution that's the limits when you have a very slender helix in a Stokes flow. It converges to that integral. And then you do the same thing. You place one sphere there. You look at the flow from the helix. That's going to move your sphere. The sphere is creating your stress-set disturbance, and you integrate that disturbance flow over the sphere, over the helix again. And so to the first order, numerically this time, you get the change. And if you do that, you can actually start to have quantitative comparison for all these shapes of helix for exactly the experimental results, so the force and torque. And I would consider that satisfying fit for like having no fitting parameters in your system. So you get the same idea as before. Both the drag and the thrust are increased, but the thrust is increased more, so you go faster. And one cool thing is that here you can look really at what happens depending on where you place your sphere. And for a cylinder, as you mentioned, unless you look at the end effects, it's always in the same direction. It's always increasing helical efficiency. But that's not the case for a helix. So for a helix, if you put the sphere in the blue region, the helix is faster. If you put it in the red regions, the helix is slower. I don't really know why, but there is that helical shape outside the helix where it's actually negative to have a helix there. And therefore, all these effects are just an average between some positive region and some negative region. And that's really the same for all the geometries that they looked at. So I think the key takeaway of that is this is basically the simplest complex fluids type of system you can have. And actually, it's already pretty complicated. So yeah, complex fluids are always going to be very complicated, as the name shows. So they were, yeah. And yeah, so basically there, we can do with the simulation, we can also simulate a free swimming helix, and we do get a speed increase that's about a third again. So that's coherent with their swimmer experiments. I don't know if it will be quantitative yet, but that's at least the idea. And you can get that from fairly simple things. So just having a line integral of solutions of Stokes equation, that's easy enough. But even here, we have very local effects that are highly non-trivial in the system. And so, of course, if you take a system where you have boundaries, and then you have a fluid that actually has polymers, and then you put an external flow, it's going to be way less easy to have these quantitative models. And it gets a bit more messy. And as a theoretician, so all of these having sort of bouncing steric interactions is a generic feature. You give me another shape, I can predict where it's going to go. The complex fluid, these ideas of line integrals and things like that, also very robust in terms of what people are doing in theory. What I'm doing for this last problem, much less robust. It's really, you have too many things and you need to think about what is the right effect and what is the minimal expression that you can write for that effect. And that's that. So this is a more recent project at UPenn. And it has to do with the fact that if you look at bacteria, you put them, bacteria, but also spermatozoa and a lot of flagellated microorganisms. If there is an external flow, they're going to move upstream. And so that's a big deal because it means that if you have a urinary tract infection, you're going to end up in the hospital for a kidney infection because the swimmers go up. It's also positive for some cases. If you have spermatozoa, it means that they can use flows in the cervical tract to actually go for fecundation. So that also is important. And it also means that in a hospital, if you have some infection near you and you have a drip, maybe the bacteria can go up the drip and contaminate the source and reproduce there, and that's actually bad for you. So it's an important phenomenon. It's a very generic effect. So I'll come back to why, but it's a purely passive effect. So it's not that the bacteria have to do anything to go upstream. It's just you put them there, you put them near a boundary, and they're going upstream. And the big question was, how does that change if you have a biological fluid? So is it different if you think about saliva, if you think about mucus or things like that compared to water? So all the experiments were done in Paulo Aracia's group. The student was Brian Torres Maldonado, who has just graduated now. And what he looked at was this microfluidic chamber. So the snake thing is just so that you have a good control over the flow and you don't have any disturbance. But what we looked at is the central chamber, and in particular, that red region. So you push a flow here, you put bacteria in the chamber region, you look at them near the boundary, and you look at how much they're going upstream or downstream. And you do that in water, which had been done and studied already. And then you do that with xanthan gum. It's a food additive. So if you look at lists of ingredients that are super long, you have xanthan gum usually. And for simplicity, you can just consider it as like very short rod-like polymers. So no flexibility, which means no elasticity. And because of thermal noise, if there is no flow, the sticks are in all directions uniformly. And then if you impose some flow, they're going to align with it generically. So that's because of hydrodynamic interactions. And it's easy to picture why having things that are aligned makes it easier to flow. So basically, it's a fluid where the more you shear it, so the more you have flow gradients, the easier it is to flow, so the lesser the viscosity. So for those of you who like rheology, this is the type of plot that we get. It's a shear sealing fluid, which we modeled by Caro fluids, but really the only thing you need to have as an intuition is the stronger the gradients, the easier it is to flow.
And that's a polymer that the bacteria don't.
Yes, so Paolo checked that. So these are the experimental results. These are just pictures. There is no difference between them. But on top, you're going to have the Newtonian fluid, so that's water. But they also tried Fickle and things like that to make sure that it's not just a change in viscosity without any complex effects. And then the shear sealing one, and here it's results for trajectories at the same flow rate. And this is cool experiments because the results are super different. First, the color. In water, the trajectories are in blue, which means that they're getting washed out by the flow. So they're going in that direction, they're going downstream. Whereas in the shear sealing one, they're in yellow and red, so they're moving up. Second thing you see is the shape and the drift. In the Newtonian one, you see that they're sort of going up. I'll call it going to the right in the top. And they're also doing loopy trajectories. The shear sealing one, they're noisy, but they're just upstream. Okay, so this is really what we're going to try to explain, and that's that. But you can be more quantitative. You can just check whether they are indeed going upstream and downstream. That's the case. Very big difference between the two. And you can also span, yes. Not exactly. It's like 1 and 1.2 in the shear sealing fluid, but this does not explain any of the results. And in particular, it doesn't explain the second effect that I'll show after. So there is a bit of a velocity difference in the shear sealing fluid. They're a bit faster, but it's not what we're seeing here.
That's the velocity in the local flow is dry.
No, so that's with the local flow. Yeah. And we're just rescaling usually by the swimming velocity. So it's V over their swimming velocity without flow. So the curve that we're interested in are these two. So first, this one, so here it means just stronger flow. So we're just looking at the shear rate, which is how strong the local gradients are near the surface. And this is their velocity in the flow direction. So if they're in the top region, it means they're just being washed out by the flow. And if they're in the shaded pink region, then it means that they're able to swim upstream. Red points are the shear sealing, so polymers, and blue points, no polymers, so empty dots. So one thing you can see is if you have a very strong flow, well, they're just going to be washed out. They cannot withstand that. But if you have weaker flows, and that is flows that are of speeds that are comparable to their swimming speed, in the shear sealing fluid, they do manage to move upstream. And this is dramatically more efficient than in the Newtonian case. They can do that at a much larger range of flows. And so that's like a factor of 10 between the two. And they can also go much faster. So they're much more efficient at swimming upstream and therefore much more efficient at contamination in the biological fluid. Mostly bad news. The other thing that you can check is their lateral speed. And that really cannot be explained by any change of velocity. This first plot cannot either, but this one, you can see that in the Newtonian fluid, the more you crank up the flow, the more they tend to swim to the right as a net drift. Whereas in the shear sealing fluid, they swim a little bit to the right, but that's a very weak effect and it plateaus quickly. And so this is basically the system that I've been trying to understand. Yeah.
So this is with the shear thinning fluid, is that, I mean, do you also get things with other types of complex fluids, and is shear thinning like the correct one to think about in biological systems?
Yes, okay, so you get that. So it turns out that I was working with Paolo on this, and then I talked to Arnold Matthijssen, who's in the physics department, and he was working with Yiling Wu's group in Hong Kong on the same experiment but with DNA. So DNA is a long polymer, extremely elastic. So it's shear thinning and elastic. They see the same thing. And what we were able to work out, because we had these two sets of experiments, so it was a good coincidence that they talked to the same people, basically. We were able to work out that shear thinning is a very important effect, and it's kind of the dominant effect, except that elasticity, and I won't talk about this here, but elasticity basically starts playing out here, and it's going to make the range at which they're able to swim upstream even larger. So their shear thinning and elasticity seem to go in the same direction for both these fluids, and their effects are distinct in terms of what they're doing. But here it's only shear thinning, so we know that this is going to happen. And shear thinning is something that's very generic. It happens anytime you have particles that are non-spherical in a fluid. So it's, in most biological fluids, they're shear thinning. But elasticity is often thought as the dominant effect in these fluids.
And just to clarify,
Yeah.
left and right is defined relative to the orientation of the axis of the swimmer relative to the flow.
To the flow. So
So there
you have like, this is up and this is left and right. So it's plus y and minus y.
they visualize.
Yeah. So to me, it helps me with left and right, especially because then it's very obvious, probably to a lot of you, why I talked about these chiral swimming with boundaries. If you're swimming in a circle, you're basically always swimming to the right. So yeah, spoiler alert, that's going to come back a lot. So yeah, this is how to see the problem as a theoretician. You have a swimming speed, V0. And yes, there's a bit of changes in swimming speed, but in the model, I'm going to take it constant. You have a swimming height, H, and that matters because if you're really high, you're going to get washed out the flow because the flow has like a gradient shape. You can extract the height from the data at very high flow rates. And it's the same in the shear thinning fluids and the Newtonian fluid. You also expect that because anything that's going to lift you up usually involves elasticity. And here there's no elasticity or negligible. And then you have a tilt angle and a lateral angle. And basically, if you know the angle, the angular position of the bacterium, then you know where it's going to go. It turns out that in the shear thinning model, theta is going to be constant. And so I'm going to completely ignore it and focus on the lateral orientation. But since you already mentioned elasticity, when you have elasticity, basically, what happens is theta increases because of that elastic lift, and that makes you more sensitive to flow gradients, and therefore more efficient. That's basically, but I'm going to ignore all of this. So you have constant height, constant swimming speed, constant tilt angle, and what we're interested in is how you're oriented with respect to the flow direction. So if you think about first a bacterium near a surface in a Newtonian fluid, so in water, it's swimming, constant speed. But in order to swim, it is rotating the helix. So that's what we talked about before. And it has to be force-free because it's small and at low Reynolds number. So if you're in the micron size, you have to be force-free. So if you think about that equation, mass times acceleration equals sum of forces, you have no acceleration, no mass. So it's the force system that has to be 0. And you're also torque-free. So you're rotating the helix, and torque-free means you have to counter-rotate the head. So if you do that, and you do that above a wall, you see that there is some symmetry breaking happening because of the presence of the no-slip wall. And it goes in the intuitive direction. In the lower part of the sphere, in that layer here, you're going to have stronger gradients and therefore more friction. And therefore, you're going to roll. So that sphere, when it's rotating at some speed, it's going to induce a translation, which is basically a wheel. So 2 translations means constant reorientation, means swimming in circle. And then the last ingredient is how you respond to the fluid. So you have a flow gradient. You can think of the head as something that's really close to the surface, not feeling much flow, basically acting as an anchor. And you can think of the flagellum as something that's freely beating. So this is the kind of thing where you're just building minimal expressions. So what I did is you take a sphere that's held at a constant length and you look at how it responds to the flow. And if you just look at the phi direction, it really has the simplest form possible, a sine phi, so purely aligning downstream, and some coefficient that is proportional to the strength of the flow. So this is the weather vane effect, the metaphor being that the flagellum acts as a weather vane. And it means that your swimmer is just going to go upstream. But it's going to go upstream while trying to swim in circles. So if it does that, initially, what it's going to be doing is basically swimming in circles, then swimming in circles, but slightly responding to the flow. So loops that are a bit deformed. And then fully responding, so no loops anymore, but still swimming to the right, so drifting to the right. And you add noise to that, and basically you get a smooth transition from loopy trajectories to just having a strong drift to the right, which is really what you're seeing here. Now, the question is, how is that affected in the shear thinning fluid? And the big thing you have to go back to is what happens when you have a sphere or a cylinder that is rotating above an oceanic wall in a shear thinning fluid. And it turns out that there was a fairly recent paper, so in Ocean Pack's group in 2021, where they looked at simulation of the system. So we redid these simulations. You have a cylinder, you have a shear thinning fluid. Again, more gradients means less viscosity. And so this is the local viscosity map. And it's super nonlinear, it's very complicated. There are no power laws that you can extract. The asymptotes as dip regime don't work, but you get a high shear region that's just created by that rotation. So at the bottom of your cylinder, you have a lot of shear, which before created more friction, but here you're decreasing the viscosity, so less friction. So you have shear thinning induces a drift in the counter-rolling direction if you do a force balance. So depending on your parameters, you can be drifting in the opposite direction or not drifting. And it really depends on how shear thinning you are, how high you are. But there is a generic effect that compensates for that chirality. If you go back to the experimental trajectories, it shows massively. So initially, we didn't even look at what they were doing at zero flow, but they're almost going in straight lines, or they're doing very, very big circles. So the other ones are doing sort of small circles because they're looping. The shear thinning ones are doing big circles or straight noisy trajectories. Then if you add an external flow, it becomes even more complicated. But roughly, a second effect is that flow is just having to go past an obstacle. So high shear region, low viscosity region at the top. And you have two competing effects. So you have one regime where you're going to have the obstacle effect that's dominating. And this is when you just have the rotation. So this is what we had when you have no external flow. And if you add an external flow, you have a whole range of things that can happen. And in particular, you have a shear dominated region where you're translating in the rolling direction again. But it turns out that for our parameters, we're going to be safely in the counter rolling region. So we're always going to be doing bigger circles. So these are the quantitative simulation. The way I introduced that in the model is very non-quantitative. I have one constant, which is what happens with no flow, and then some alpha parameters, which is saying that if you add a flow, you're decreasing that effect. So this is why I meant that in this system, you really want to go for the minimal expression that you can have. But it does work. That first constant, it turns out to compensate. You can get it from the experiments. It compensates exactly on average, the chirality. So they're swimming in straight line with a slight bias when there is a flow to the right. And so that's that. The weather vane effect is not affected. We also did COMSOL simulation that tend to show that. And so you end up with some equation, which has some very simple form. It's called Adler equation. And that it was introduced for when you have two oscillators and they have their own phase, but they also have a tendency to synchronize. And so what happens is you have a constant, which is basically their own phase. So that's their tendency to do circles, but then they tend to synchronize with the flow. And then they're not doing circles, they're in phase with the flow and they're not doing loops anymore. And you have a very nice transition here that is basically controlled by how big your circle is. So if you're doing a very big circle, it's really easy to synchronize with the flow. And so all of that just gives you back the experimental results. You have the weather wave effect is the fits for the two Newtonian curves. And then the alpha fit is basically your response to the flow is basically going to set that height. But roughly the effect is if you're not trying to swim in a circle, it's much easier to respond to the flow. That's just that easy. And that also explains why your trajectories move from being loops to being straight lines. And so we think that this has very big implications for how contamination happens in biological fluids because it's a very dramatic effect. And we've been starting a collaboration with the dental school to see how that applies to saliva, even just the trajectories. And so we're working on that. But basically the takeaway is you're better at doing things if you're just noisy and not looping and being advected downstream. And so, yeah, this is basically a picture of where we've been on my sort of phase map of things. So boundaries, complex fluids, and then putting everything together in that last part. And with that, I would like to thank my collaborators at UPenn. So Yoshiro, Arnold, and Paolo with whom I've been working. Then the two first part were done at Cambridge, and Florence, Roberto, and Andres for the experiments here. Thank you.
Couple of questions.
You have mentioned that with collective behavior, that a lot of these effects might change. Would you imagine that maybe the upstream flow could change in the complex fluid with multiple of these helical polymers?
So I think the range at which the interaction would happen would require to have a very, very high density. We're not seeing anything happening when we increase a little bit the trajectory, so I don't think it would be huge, very obviously, although, yeah, maybe actually, yes, I don't know, is the answer. Because for bacteria, we don't see anything, but for spermatozoa, they're known to swim in bunch, so they're attracted to one another, and they swim in like small groups of maybe 3, 5. So, and they synchronize there, so there I would expect that it would matter. So maybe experiments dependent, but yes, it might.
So maybe in the interest of time, if anyone has a question, we can send them afterwards, but thank you so much.
Thank you.
Long